Hello everyone, welcome to MedTwins. This is a video on the eye physiology and the visual pathway. So let's start off this video by talking about the anatomy of the eyeball. So the eye is made up of three important layers. The outermost layer, that's made up of the sclera, the white portion of the eye that you can see here. The cornea, which is this area here, which is important in the refraction of light. However, it cannot really change its shape. Then there's the middle layer, which is made up of the choroid, which is this vascular area that you can see here. Also the iris, which is made up of two important muscles that we'll talk about in a second. The iris is what gives the person the color of their eyes. And also the pupil, which is the hole in the middle of the iris that lets light pass through. And an innermost layer, which is the retina. The purpose, which you can see here. The purpose of the retina is to transform all the light into nervous signals, and which can be then taken to the brain via the optic nerve. Before we move on, I'd like to point out some important structures in the eyes. They're the ciliary body, which produces the aqueous humor of the eye, but also contains the ciliary muscles, which can be seen here and here. And these attach to the lens of the eye via the suspensory ligament that you can see here. This is important, the accommodation reflex, which is basically focusing the light on a near object or on a faraway object. So if you want to look at something that's close to you, for example when reading, the ciliary muscles will constrict, making the lens rounder, which will help focus light on a near object. However, if you want to look at something that's far away from you, these muscles will relax, which will make the lens flatter, focusing light on faraway objects. There are also the two muscles that make up the iris. These are the radial muscles, which help in dilation of the pupil, also known as mydriasis, and the sphincter muscles, which help in constriction of the pupil, also known as miosis. There's also the fovea, which you can see here. The fovea is important because it's an area of high resolution in viewing, and it has a very high cone concentration, which we'll talk about in a second, but it's very important for vision. Now, let's talk about the physiology of the eye and also the microanatomy a bit later on. So, the visual pigment in rods and cones, which are both of the cells in the eyes, is an 11-cis retinal bound to an opsonin protein. Whenever light hits the eyes, especially these special eye cells, the rods and the cones, it converts this 11-cis retinal to an all-trans configuration. This is very important because when it's converted to an all-trans configuration, the opsonin molecule is released, and this, in the process, retinal is converted to retinol. Another important factor is that the release of opsonine molecule from the all-trans retinol causes bleaching and loss of the ability of that specific cell to absorb light. In order to regenerate the 11-cis retinol, the all-trans retinol is transported to the pigmented cell layer, which we'll talk about later on. However, the cones have an alternative mechanism, which we'll also talk about later on. From this slide, I included an image of the entire process that was just described. However, the most important step is this one, that light converts the 11-cis retinol to an all-trans configuration. If you want to spend your time memorizing this, you can pause the slide and take a look. So during darkness, photoreceptors are depolarized because of high cyclic guanine monophosphate, cyclic GMP, that keeps cation channels open. So this can be seen here in the image, where the high GMP in the cell will keep the Na channels open. This is happening during darkness. When light enters the eyes, the photoisomerization of rhodopsin will activate transducin. The photoisomerization of rhodopsin is what we just described, from the 11 cis formation of retinol to the all trans configuration, which will release the opsinin molecule. And this will activate transducin. Transducin is important because it will activate phosphodiesterases to reduce cyclic GMP inside the cell. If we reduce cyclic GMP in the cell, this will cause the cation channels to close. So the Na channels will close once the cyclic GMP is reduced in the cell. Closure of these cation ions will lead to hyperpolarization of the cell membrane, and this will lead to a reduction in the release of neurotransmitters from the cell. Now let's talk about the microanatomy of the retina, which is really important as well. So the retina can be divided into 10 layers. And down here is a pigmented epithelium, which we mentioned before because it is involved in the restoration of the all-trans configuration of retinol back to the 11 cis retinol formation, especially in rod cells. However, cone cells can have an alternative metabolism to do that, and we'll talk about that later on. Then here are the photosensitive cells, the cones here. They have this little cone at the end. And the rods, which are longer and can be seen here. These can then have connections with bipolar cells, shown here. And then these bipolar cells can relay this information to ganglion cells, which would then get together to form the optic nerve. Note that rod cells can have, like a bipolar cell can have a connection with several rod cells, but usually the connection between a cone cell is more direct to a bipolar cell. Of course, bipolar cells can still be connected to several rods, but usually it's a more direct connection, whereas with the rods, it connects to more cells. The bipolar cells connect to more rods. Now let's talk about the individual photoreceptor cells, starting off with the rods. So the rods are mostly used during darkness. That is because they only have one pathway to regenerate the 11 cis retinol, which takes longer than in cones. So they can become overly saturated in light. However, during darkness, where there's less light, they're usually used. Because we only have one pigment for rod cells, we cannot distinguish color with them. And as mentioned earlier, many rods can converge into one bipolar cell. This causes a high receptive field. However, there's low resolution. When it comes to cones, they're used during brighter conditions, such as during the day. They have quicker regeneration of 11 cis retinol because of a secondary pathway using Muller cells. They can distinguish color because of three separate pigments that they have. The S cones, which can identify the color blue, M cones, which can identify green, and L cones, which can identify red. Cones produce a higher resolution images because of little convergence with bipolar cells when compared to the rods. Now let's talk about the visual pathway, which involves the optic nerve, optic chiasm, and optic tract. But please keep in mind that this is just one of the visual pathways. The information from the retina can go to other parts of the brain. However, only the main one is going to be discussed in this video. Now let's talk about your visual field. So to start off, it is very important to realize that we have a temporal portion, which in this case would be to the left, and the nasal portion, which is towards the nose. Temporal means towards the temporal lobe, and nasal means towards the nose. So whatever you see in your left temporal visual field, that light is being absorbed through your nasal retina, in this case here. Whatever you're seeing here on your nasal visual field of the left eye is being absorbed by the temporal retina of the left eye. Same applies to the other side. So whatever you're seeing in your temporal visual field of the right eye is being absorbed by the nasal retina. And whatever you're seeing on your nasal visual field is being absorbed by your temporal retina. So then this information is transmitted through the optic nerve, and it's very important for us to realize that in both nasal sides, so this side and this side, the information from these sides cross at the optic chiasm here. This means that information from the left eye's nasal retina, which is this area here in red, crosses over to the right hemisphere of the brain, and it will synapse in the lateral geniculate nucleus. This also means that the temporal retina of the right eye won't cross over, so it will remain on the same side of the brain, and it will go to the lateral geniculate nucleus. The same thing will happen to the other side. So the nasal retina here will cross over to the other side of the brain, so to the left hemisphere of the brain, while the temporal retina of the left eye will stay on the same side, and both of these fibers will synapse at the lateral geniculate nucleus of the left hemisphere of the brain. After synapsing the lateral geniculate nucleus of the brain, these fibers will be carried by optic radiations to the primary visual cortex in the occipital lobe of the brain. It's also important for us to notice that the fibers here on the left lateral geniculate nucleus of the brain will be carried by the optic radiations to the still on the left hemisphere of the brain to the primary visual cortex. Same thing on the other side. These fibers that synapsed with the right lateral geniculate nucleus will be carried by optic radiations to the primary visual cortex on the right side of the brain. Now let's talk about some lesions in the visual pathway. So depending on the area of the visual pathway that the lesion has occurred, the patient will have different vision patterns. So if a lesion occurs at A, which is the fovea of the eye, they would lose on that eye the center of their vision. If a lesion occurs at B, as we can see here, the person will lose all of the information coming from the right eye, at least when it comes to vision. However, if a person has a lesion at C in the optic chiasm, remember, this is where information coming from both nasal retinas cross over to the opposite hemisphere of the brain. So they would lose all the information coming from both nasal retinas, seen here. they lose information from here and from here. And remember, the visual field is the opposite to the retina. So they would lose information from the temporal visual field on both eyes, seen here as bitemporal hemianopia. Now, if a person has a lesion at point D, seen here, they'll have what's called contralaterous homonymous hemianopia. And this can be explained if we follow the red fibers here. So the red fiber that crosses through the optic chiasm is on the left eye on the nasal retina. So this would receive information from the temporal visual field of the left eye. So the person would lose information from the temporal visual field of the left eye, as well as here. So from the temporal retina of the right eye, which would translate to losing the nasal visual field of the right eye. When it comes to lesions in E, here, J, F, and I, as seen here, these are lesions to the optic radiations. And in K, there's an interesting pattern, and that can usually be explained by the double blood supply of the macula, which is the center of the eye here. It receives blood supply from both the PCA, posterior cerebral artery, and the middle cerebral artery. So this could explain how it can be preserved in certain lesions. Thank you very much for watching this video. If you enjoyed, make sure to like and subscribe. If you find anything that you think is inaccurate, please let me know. If you're interested in looking at any of the images, I'll leave a link down below to where I found them. And yeah, thank you very much. I hope you all enjoyed.